All right, hey guys. Thanks for having me. I'm really excited to be here to give you guys a talk. Today I'm going to talk a bit about my work, which is a generative model for single-cell RNA sequencing, where we do nonlinear manifold learning to learn like latent embeddings, and also just a little bit more generally about different dimension reduction techniques that are common in single-cell RNA sequencing, because it's a really important part of the pipeline, and people use a lot of different models, and I think it's good to have an idea of what's going on with each, and what are the pros and cons for each of them. So single-cell RNA sequencing has a couple of analytic challenges. You need it. It's a really important novel technology that's enabling a lot of new insights. So like cell states, cell types, developmental pathways, these intrinsic, extrinsic, and tissue scale insights. But it's difficult to analyze some of the data that comes out because we don't necessarily have a lot of prior knowledge. We don't know what's really inside the data. And there's a lot of dropout. There's a lot of technical noise. You get doublets where two cells will be encapsulated in one. And there can be batch effects between different samples. And so we want to develop a really robust, low-dimensional generative model for single-cell RNA sequencing to do this dimension reduction that people used in downstream pathways. So why do we care about dimension reduction? Why do we care about manifold learning? There's a lot of downstream applications, and it basically serves as the first step in any of the analytic package you're going to use. So the first thing is just a normalization. You kind of regularize some noise by throwing it to a latent space and reducing the dimensionality. You can try to align different manifolds from different experiments and use that to kind of correct between batches, which is something that like Satija and Marioni have worked on recently. You can use your lower dimensional space to then learn some similarities and use that to then impute dropouts or missing cells or just remove some technical noise and use that for maybe a better downstream analysis with a more dense matrix. You can use it actually as the basis for your pseudotime estimation. Pseudotime in single cell refers to if you have a single batch, but you believe that the cells come from different kind of temporal developmental stages, assigning labels of which cells come from which order. And then obviously visualization, if you've done any work with single cell or read a single cell paper, I'm sure you've seen one of these beautiful t-SNE plots, but it's very important to know how do these visualizations come around and what can we actually interpret from our low dimensional visualizations. You can use it for density estimation, which is then kind of feeds into experimental design at the bottom, where you can say, okay, I know I've sampled this part of this space. I know I have cells in this marker region or this kind of behavior region. I want to maybe move my experiments to sample a different part of the space. Or just say I can get insights from the space I have. This is kind of the area of this biological space I can actually make inference about. You can use it to then kind of define clusters and use that for differential expression, looking at expression gradients across cells. And depending on the kind of generative if you use a generative model, you can learn uncertainty about your mappings. And you can learn where am I confident in my predictions, where am I not confident. So we want to build a generative model that can do all these things. And we especially, because we have noisy biological data, want to incorporate some robust features. So robustness just means that given outliers, as you go to Indies, you can discard those outliers and still learn the true model. So one thing we use is a student t-distribution as our error model. So here you can see a figure where if I have like a true underlying manifold, we simulated this data, and we see these noisy red observations. If I use a normal error model to try to fit this manifold, it kind of overfits and is really spiky towards the experimental data that we see. But if I use like a t-distributed error model, it learns a much nicer and smoother kind of manifold. Yeah?
Shouldn't the performance of each of these depend on what model was used to generate samples?
Absolutely. This was generated using a t-distributed error model, and so it looks better. Whoop-de-doo. But in real data, actually, so there's these papers from Jailenki and Van Houten, I think 2010, 2011, where they use time series data for biological gene expression, and they find that using something like t-distributed error does a better job of smoothing out your time curves over time and doing future prediction. So because we're in an unsupervised setting, we don't know the true model from which it's drawn, we believe that a t-distribution and a robust t-distribution makes more sense for noisy technical biological data.
So you want fatter tails?
Yes.
How many degrees of freedom?
So we set the degrees of freedom to 4. That's based on previous literature as well. But yes, you're right. It depends. If you draw something from a normal model, a normal error is going to work better, right? But when we're working with real data and we're applying a generative model, we don't really know. And then return kernels, which are kind of non-smooth kernels that can be used in modeling. I'll talk about a little bit later when we go to the full model. Actually, next slide. So just to go over quickly what our model is, we kind of use an adaptation of the Gaussian process latent variable model. Have you guys heard about Gaussian process before? All right, so everyone's familiar with that. And so, oh, not everyone. Okay, so I'll start from the fundamentals. So a normal Gaussian process if I observe some data like this, and then there's some noise around, a Gaussian process is just a distribution over functional space. And it tries to say, given some inputs, let's say t, and outputs y, what does it believe the function is over, t to y? And it does that by saying that the probability of every point in y, so it thinks about all the probabilities of every instantiation at the same time, given t, is a multivariate normal with some mean and some covariance kernel. And the important thing is that this kernel function, this covariance, is basically, if I have n points, an n by n matrix, where each entry represents how similar the time is to each other, right? So it kind of learns a kernel function between distances. Does that make sense? And then projects that using a multivariate normal. And it depends on what kind of kernels you'll use. So the general kernel people use is an RBF kernel, which is so this is kind of a supervised Gaussian process, really quickly and really dirtily. But a Gaussian process latent variable model is similar, except we don't observe t here. We don't know this. We have a high-dimensional projection, high-dimensional vectors, high-dimensional samples, and we assume that these high-dimensional samples are actually drawn from a Gaussian process of lower-dimensional latent space, right? And so the model we can see up here, we say there's some latent variables x that we say are drawn from a normal with identity variance. Then the actual manifold in higher space, the noiseless instantiations, realizations, are a Gaussian process of these lower dimensional observations. And what basically happens is that because you have this high dimensional space with lower dimensional space, all using the same Gaussian process, the same kernel function, you share information across the dimensions. Yeah?
So in essence, the sparsity of the Gaussian process's covariance matrix is what determines the dimensionality of the lower latent space?
Yeah. Basically how many terms do you need to actually properly represent the covariance, which will be the sparsity of the lower dimensional space.
So, in the equal question, there is an order of those variables, right? But here, what is the order?
There isn't an order. Do you mean like here there's like a temporal order, right? There's some, excellent. Yeah. So that basically in the unsupervised version, you're learning both t and the kernel parameters at the same time. So hypothetically, the ordering could shift based on which kernel parameters you use, right? But again, this is a representation latent space. If it's the same, if I double everything in double length scale, it should be okay. Someone else have a question?
So just to be clear, there is no, it happens that in one dimension there is an order.
Yes.
Real numbers.
Yes.
But the kernel doesn't use the order.
Yeah.
It uses just the real numbers.
The kernel, yeah.
Similarly, in the plane, which that function lives on top of, you can talk about the distance between points and have the
Exactly.
kernel.
Yeah. And again, depending on how you define your kernel, most kernels are symmetric. So actually, time is a common one that's used for, but it could be literally any input covariance. And so what really matters is the distance and then the kernel function of the distance, right? Now you can design, people have designed forward-looking kernels for time series modeling and stuff, but for an unsupervised, we aren't going to worry about that kind of stuff.
So you always give dependent distance by the distance between those points.
Yeah, yeah. And actually, we use something called the automatic relevance determination kernel, which is a little bit of fancy kernel parameterization, where we actually say that each dimension in latent space has a different length scale. And we learn different length scales for each dimension. And what that does is that if you set your latent dimensions to like some conservative high estimate, based on those length scales, you can determine which dimensions matter more, have more significance in the kernel, and use that to actually learn and pare down the dimensionality of the data, which a lot of models right now don't do. So,
Just to unpack that a little, can you
Yeah.
maybe elaborate on where the length scale is in this kernel and
Yeah.
what the length scale means in terms of the kind of functions that you might sample?
Absolutely. So the length scale here is this L function here, right? Which says that given the distance between the two, how much do I reduce that distance by, right? You can imagine that if I have, say, a couple different dimensions, then we define some sort of different operator. We'll call Q the dimensions. Right. So now imagine we have like a multivariate kind of situation, right? So I have Q1 and Q2, and there's some observations Y, right? There's no reason to believe that, if this was an observed or unsupervised, in the supervised or supervised setting, that the distance in each dimension is going to be the same, right? If you're modeling something with two inputs, you do linear regression, they have different coefficients. Similarly, use a Gaussian process, they might have different length scales, right? And so length scale basically determines, is it going to look like this, your kernel distance, or is it going to look more like this? How spread is that kernel function, right? So we're all happy. And then you understand that I can have different length scales across dimensions. I can learn the length scales using maximum likelihood estimation to figure out which dimensions matter and use that to pair dimensionalities. Because you can also look at 1 over L squared as this weight term. And so things with lower length scales will have higher weight. Those dimensions matter more, right? And so we also introduced these Matern kernels. So I'm trying to figure out. Yeah, sorry, my bad. Oh, do you have a question? No, just. Okay, yeah. Is there an eraser? Or?
This might work.
Cool.
If it doesn't work, this should work better.
All right.
If that doesn't work, add a bit of water.
Cool. This is fine.
Also, today only, if you ask the question,
So most unsupervised GPLVM so far have used this RBF kernel, but in a lot of supervised settings, this imposes infinite differentiability and infinite smoothness on your manifold. And we didn't necessarily want to stick with that assumption. So if we say R is kind of our length scale ARD form. Oh yes, writing big. Across M different kernels that we sum up. Then we can add these Matern kernels, which have different levels of differentiability. So there's the Matern half kernel, which is differentiable once. No, not in the Matern setting. Yeah. Oh, but it is absolute value. Yes, that was one of the lines. So these kernels have been used in unsupervised time series, including with single cell and with lots of different applications to kind of model unsmoothness, and we integrated this into the unsupervised model so that we can learn maybe different types of manifolds that may not be properly captured by the smooth Gaussian kernel. And then at the end, we use a student t-distribution with 4 degrees of freedom, as mentioned earlier, based on previous biological work, to model these heavy-tailed, really noisy expression patterns that we see from RNA-seq count data.
So,
yeah.
The notation y, where does x fit in there?
So x is our latent variable, right? We have some noiseless kind of pure observations in high-dimensional space F that are a Gaussian process of 0 and k and n, which is the kernel between all the x points. And then, so that comes here, right? So we do each of these kernels across each of the points, sum them up, use that to do a GP draw, and then have some noisy observations afterwards. Does that make sense, where x is coming? So x comes from here, you compute the kernel, do the draws, and then have a noisy observation. And if you guys are really curious, we put the manuscript on bioRxiv like 2 weeks ago, so you guys can go read about it. And it's a bit more about the model. So this.
Go back.
Yeah.
Okay, so you wrote down these 3 terms instead of, you have an RBF kernel, which
Yeah.
would encourage
So we use actually
smoothness in your function.
RBF, this, and this.
And you take some of these,
Yeah.
you learn coefficients
Yes.
as well on these. Can you motivate a bit why you have 3 additional kernels
Yeah,
here?
we want to be able to be very general and to almost any kind of manifold structure there is.
What's the difference intuitively between the half, between these 3?
It's different levels of differentiability and different levels of smoothness, right?
Can you draw them as a function of R? Like they have like different spikiness.
Yeah, they have different spikiness. So like the half is like this, and the 3/2 is more like it's a little bit smoother at the top, and the 5/2 is like a little bit smoother, and then like the Gaussian is like a totally smooth.
Right.
And so they have different levels of pointiness up here.
Right. And then sort of the surfaces or whatever that you end up sampling from your GP, there's some relationship between the smoothness of this kernel at the origin and kind of what they look like everywhere.
Yeah. So this is the least smooth, and this is the most smooth. If that makes sense. So we're trying to just give different levels of smoothness.
Right. So the very last step is what connects your latent representation back up to the original observed expression data. So that's a map that's actually going from 3 dimensions. Is it a surface plus noise? So you think of these as 3 dimensions, or you?
Yeah.
Okay.
So this is in gene space plus noise. Well, so F here is also in gene. This is in gene space, right? So this would be 3 dimensions, right? F is kind of this kind of latent variable that you use as part of modeling that says like, all right, if I have just perfectly drawn where the cell expression lies on the manifold, what would it be, right? If there was like no noise at all, no like technical experimental noise, right? And then this is a noise term that.
So your y is in 3 dimensions?
So yeah, here let's say there's like a 2-dimensional latent space, right? This is a 2-dimensional surface, right? But we see a 3-dimensional manifold, right? So x is n by 2, f would be n by 3, y would be n by 3.
F of x. So f itself is a map?
Yeah, f is kind of, okay, so let's just bring it back to 2D real quick. We have these noisy samples we see in reality, right? You know, of t, right? Now, if this is a real just function, right? Hypothetically, any value of t should only map to one value of y, right? So it's like this, right? So this is your f, right? But then because we live in a noisy world and we're doing like a generative model, we allow for some error term. And so this is kind of, right? So in the same way with the unsupervised setting, we say that for a latent space, there's some noiseless higher dimensional function where it should lie, and then we observe a noisy representation of that.
So f maps the latent space
into high dimensional space.
space of observations.
Yes.
And X are the locations in the low-dimensional space.
Yes.
And then they get mapped to the high-dimensional space plus some noise.
Plus some, yes, exactly.
So this might be redundant, but is it correct to just think of it as a regression equation where y given x is equal to f of x plus some?
Yes, yes. And f is this Gaussian process, nonlinear.
Your own f is a Gaussian process.
Yeah, yeah, exactly.
F is what would have been the line. Now it's a wiggly thing that comes from a Gaussian process.
Exactly.
Yeah.
Cool. Yeah, you guys got it.
Okay, so in your visualization, the plane, the xy plane is our x, and
No,
drawing
here,
the graph of y.
here x, y, and z are all up. This is all 3 are y.
But
Don't worry too much about this. This is just a
Okay.
pretty graphic.
yeah, I thought you were drawing f.
No, yeah, no.
That's why I'm.
So this would be f technically in 3 dimensions, right? And then
Couldn't this reasonably be a picture of f that goes from 2?
it absolutely
not
could
what
be.
it is.
It absolutely could be, but
Okay, that's
it's
not what
not.
it is.
And you're right. Maybe I should label this better. That's
There's a latent
something.
plane that parameterizes this actual surface.
Yeah.
That's the X space, right?
Yeah.
That's the
Yeah, yeah.
space in which
There was some other
yeah.
latent. Yeah.
And you picked some points on there from a normal, you threw them into this thing, and then added some noise to get the red guys.
All right, now it's making more sense. And then
Sorry, that's on me.
No, it's good.
Grab
This is
it.
fine. We need to all
This is
understand
good.
this together.
and in practice, how high of a dimension? How many genes are you modeling simultaneously?
So we actually end up modeling the full gene space. We have a really nice Microsoft Azure system, so we use all like 20,000 genes. Yeah. Q we normally do anywhere between like 2 to 10 depending on the data set. And we try to do you do conservative Q and then you pare down later. You do like 10 to 15 and then you see, okay, do the length scales really matter based on this parameterization, right? So the results we have so far, actually, it turns out that like 10 normally actually does still capture a lot, but for some of them you can go down to like 3 dimensions, right? Depending on what you want to see.
I have an extra question. So your composite kernel seems to be the sum of your other.
Yeah, it's a sum of this. And there are weights. These sigma squared terms end up being weights. And also, in a way, these length scales can also serve. So this serves as weights between kernels, and this serves as weights between dimensions. So there's a meaning to each of them. So you can say, okay, the Matern 5 halves kernel doesn't matter, right? And you find that basically the Matern half kernel comes up a lot. These two improve performance a little bit, but yeah.
And what's the feasible upper bound for P? How many cells can you fit to this?
Or N, you mean?
That's genes, right?
P is genes, N is cells.
How about N then?
N, so we implement one that scales up to 1 million and does a decent-ish job. This depends on your time scale and your computational resources. So with our 16 CPU cloud machine, we can fit 1 million. And that's basically,
Good. That's like not a huge cloud machine.
yeah, and that's because basically it's more about the implementation than about the model itself. We use variational inference, which uses inducing points. So I'll just give a quick thing here. Oh, well, actually, it's 2 slides from now, but I guess I'll skip ahead because you guys are curious. We try to make this so we can learn it scalably. And so instead of using something like a maximum a posterior estimate, where we'd have to do an inversion of this covariance matrix, if it makes sense because you have a normal and you're trying to do map, that inverse comes out. We use something called inducing points, where we introduce, say, these auxiliary variables. And we basically learn the M, and we learn like the true Y's or F's at these points. And then we use that to propagate information to the other points. And so when we have to do like an NM squared time.
So a couple of questions there.
Yeah.
Are you just using those induced points with it being part of like a mean field approximation?
So it's not mean field. So this paper, Damiano 2016, kind of goes through the math really well of how it works. Basically, mean field doesn't work for the Gaussian process latent variable model because you don't actually remove some of the intractable stuff. Trying to think what the best way, but it's not mean field.
Then the second question is, since you're doing variational inference, you have some bound on your marginal likelihood of the model.
Yeah.
I'm just a little concerned that this kernel function you have might be overfit because it has a ton of parameters. So did you do like a Bayes factor analysis comparing different marginal likelihoods depending on how many terms you have in your kernel?
We didn't do that. That's maybe something I should look at. But when we do downstream analysis, which will come up a bit in the results, then we find that the matern kernels do help with downstream analysis. But it could possibly be overfitting. That's something we should definitely check for.
Yeah, I guess I had a similar question about overfitting. Once you mentioned that the one half, the most spiky one, tends to be the one that is more sort of significant or present. Again, more spiky stuff sounds more overfitty.
Yeah.
But maybe you checked whether it generalizes.
It does seem to generalize pretty well. And adding the kernels doesn't change the representations a lot. Like when you do the latent mappings, they do end up looking relatively similar with just like, basically what ends up happening is in latent space, if I add these kernels, something that kind of used to look like this before actually comes together closer, which we like, but this would be the latent space. Yeah. Yeah. Yeah, this is kind of generally what ends up happening when we add kernels.
What's your favorite way of reducing from whatever the latent space dimension is, maybe 10 to 2?
Oh, well then we use the length scales. Okay. Yeah, that'll come up later.
So did you try to use MCMC to see how the results are different?
We haven't tried using MCMC because that just takes a long time. We have a really nice implementation that we did with TensorFlow and Edwards, which is the probabilistic programming sample, but maybe sometime, if time permits, we could try MCMC and see if it's different. All right, everyone's happy? So I don't know how we're doing on time, but I was going to do a little digression and talk about some of the other models that are very common in single-cell RNA sequencing because you're probably going to see them in papers, and it's important to understand where they come from. So obviously, you guys are probably all pretty familiar with principal component analysis. It's the basic dimension reduction technique you learn. I was an engineer by training, and even we learned like singular value decomposition, so I'm sure the statisticians around are more than familiar. But just as a brief recap, PCA is just a matrix decomposition where we say that our high dimensional observations n by p come from some latent space and then linearly transformed by some factor loadings plus a noise term. And here, the noise is the same across all features, right? And we solve this using SVD. We minimize y minus z lambda. You can also put this in a probabilistic perspective and learn like posteriors by saying z is drawn from a normal 0, 1, and y is a normal to z theta, mean z theta, and some error epsilon.
Would you agree that if you do that, then the setup becomes quite similar?
It does. So actually, PCA is a Gaussian process latent variable model, but if you define the kernel as a linear kernel, just the distance, and the noise is normal, it reduces down PCA. So they are actually, you can think of the Gaussian process latent variable model as a nonlinear generalization of PCA.
PCA is literally just fit a multivariate Gaussian to the data and then project that to the eigenvectors.
Exactly. Yeah, and you find the orthonormal eigenvectors that maximize the variance across the data, right? So this is really nice because we have really good algorithms to fit it, right? It's very fast. You can look at your data really quickly. You know you're capturing the maximum variance, so you have a good sense of like what's really changing across different cells in your data. And you know what everything means, right? I know that moving like 1 unit in any dimension z is associated with an expected increase of whatever that lambda q is, right? So it's really nice. And as a first pass analysis, it works really well. But then some of the problems with PCA that come up in single cell is that the linearity assumption doesn't necessarily hold really well for biology. You'll get cells that maybe are really similar, but because of noise or because of just the way biological processes work, are kind of non-linearly distant from each other. And it can be really heavily dominated by two things. One is oversampled cell populations. Because it's trying to capture the maximum variance, it's going to be really weighted towards cell populations that are heavy. And this makes it hard to find maybe rare subpopulations that are smaller, because they'll kind of be pushed towards like the 0-0 space because of the other one's variance. And it gets really dominated just by just a lot of reads. If you have one cell with a lot of reads, it's going to look similar to another cell with a lot of reads, even if their actual expression isn't that similar. And that means that things that are just big, or cells that got a lot of reads, come together even if that's not really informative. Okay. So that's PCA. Very nice method, very cool. But biologists are too cool for that, so they all started using the T stochastic nearest neighbors embedding, right? You guys have all seen a T-SNE plot in your lives? Cool.
Now everyone has.
Oh yeah, now you guys have, if you haven't before. How many of you guys actually know how T-SNE works? All right, cool, so a couple of you. All right, cool, so this is good. I'll talk about it. So T-SNE at a very high level, you can think of as it learns a kernel matrix over your high dimensional, your observations, and then it tries to learn a latent mapping that with a different kernel would recreate that high dimensional kernel matrix, right? So you're almost like kind of matching covariance between the high dimensions and low dimensions, if that makes sense. So the actual formulas is that given some observations y in of size n, we define this P matrix where P of j at I, given I, is this kernelized based on like a diffusion process. And then normalize across all other maybe diffusion options for that cell. So do people kind of get an intuitive understanding of what this means? It's basically saying that if I wanted to move from cell I to cell J and it was working as like a random walk, what's my probability of moving to cell J of all the other possible neighbors it would move to based on this diffusion distance with this diffusion scale? And then, so this is a probability. They actually end up making the matrix symmetric for computational regions. So Pij, which would be equal to Pji, is just the symmetrized form of that, right? So this is our high dimensional matrix P that T-SNE produces, right? Then, from our lower dimensional things, from Z, we give a different distance metric. Right? So here the kernel is this kind of Cauchy distribution or 1 degree of freedom T distribution in latent space. And then they tried to minimize the KL divergence between the two. Overall. And they set everything on the diagonals to 0. So you're not worrying about the self-similarity, right? So this is the basic setup of T-SNE. We have a high-dimensional covariance kernel matrix, a low-dimensional kernel matrix with a different kernel, and we try to fit them so they look the same. So
I think you're actually fitting.
yes, you fit Z. Everything else is selected, right? So this has been used, it's very popular in the single-cell community and basically for visualization because it produces these really nice plots with really distinct clusters. There are some issues with T-SNE.
Can you say a word on why these particular kernels encourage that behavior?
Right. Basically because this is kind of a heavy-tailed versus this is a like normal smooth thing, it likes to push things together on this heavy-tailed in this lower dimensional space, right? And It likes to keep things that are close to each other similar. Basically, so the thing with t-SNE, and what ends up becoming the problem with t-SNE, is that it likes to learn clusters. It likes to find similarities. So if I were to simulate random noise and put it through t-SNE, and they have this demonstration online, you can look at it, it's like the first thing, like t-SNE problems, first link. If I simulate a random noise and I put it through t-SNE, depending on how I choose my perplexity, it's going to find clusters regardless, right? t-SNE is very prone to overfitting. And it's very sensitive actually to this parameter, this length scale parameter, which you input to t-SNE as the perplexity, which defines the entropy of this distribution, so for each cell. But basically, if I change the perplexity, if I increase the perplexity, it means that I'm okay with more nearest neighbors, and it's going to change something that maybe with low perplexity will look like this, and then in high perplexity it'll look like it's all just one cluster, and then depending on that perplexity change, I can kind of find as many clusters as I like. And then another problem that ends up coming with t-SNE is just the way it's formulated. There's no global structure to the latent variables. So moving in this direction, I have no clue what that means, right? There's no actual mathematical function relating this to this. It's just learning these similarity metrics. And you'll see that in t-SNE plots, we have one that comes up later, where I can cluster something in high dimensions, right? I'll do my hierarchical fancy clustering on my count matrix, put into t-SNE, and cluster 1 will be here and here, right? Because it's learned that, well, these cells are kind of similar, and these cells are kind of similar, and these cells are slightly more similar than these were to what this was, and it separates things that are maybe actually similar, right? Alex brought up right before talking that UMAP is like a new, fancier version of this that maintains a global structure. I don't know much about it, and I'm looking forward to learning more about it and using it, and maybe that might have solved some of the problems. But the way it's currently implemented now, t-SNE doesn't necessarily give you a very real representation of your data in lower dimensional space. Yeah. Yeah. I mean, it's definitely a very helpful tool for visualization, and it works really well. Like if you've done your clustering and you get a nice t-SNE map to represent your data in a way that the audience will find convenient, right?
I would not overstate the overfitting aspect, right? The nonlinearity of the non-intuitiveness of the distance. If you just look at the nearest neighbors graph, it captures that.
Yeah. Well, because I mean that is what t-SNE is entirely dependent on, right? It learns the nearest neighbors graph. So, I mean, it's similar, almost like a diffusion maps thing, which generally also work really well, right? You have this kernel, and then you do. No, okay, that's too much. Okay. No, I mean t-SNE. I mean, there's a reason it's so popular, right? Because it works. Yeah, I'm not gonna go here and be like, never use t-SNE. Like, there's a reason.
Never use the output of t-SNE as the input to something.
Exactly. The thing is, you can't use t-SNE downstream, right? It's great for making graphs. It's great for visualizing, but that's where it ends, right? And you have to make sure that when you do a t-SNE plot, you don't like overly convince yourself that this is exactly what the data look like. You have to remember this is a visualization that's convenient that t-SNE is produced, right?
Great blog post on how the relative size is and the relative distance is. Those all mean nothing.
Yeah, so. There's no meaning to your t-SNE plot. That's just the important thing to take away. But it's gorgeous. I'll give you that. It's gorgeous.
But it does tell you things about the data.
Yeah.
Instructive about what you do.
Oh, absolutely. Yeah. It's more about like these kernels lead to clustering. The KL divergence is just a way of, KL divergence is a similarity metric between the two, right? Yeah, QIJ is look at clusters in lower dimensional space. So what really creates the clusters is that you say that in high dimensional space, these are its nearest neighbors, right, with some strength. And so this tries to recreate those nearest neighbors, right? Now, every cell is going to have nearest neighbors, right? And so even if a cell is actually kind of far from something, it has nearest neighbors, and it's going to be in lower dimensional space, be like, oh, like there's its nearest neighbors, so I'm going to cluster it near these things, right?
I have a point to make. Like, this story looks like you cannot make clusters from Disney, I mean, apart from visualization. You can get those Z values in the lower dimensional space and use them to do some clustering methods.
You can, but that's what becomes kind of problematic is that we don't know there's no like proper interpretation of Z. There's no function proper function coming from Z back up to Y. You can have these kind of like expression patterns that look like this across Z, right? You have to be, and it's very sensitive to this, right? So it looks good, it'll give you a lot of insight into your data, but the actual latent dimensional space doesn't really have a good meaning.
Yeah. I just want to make sure that we have time for you to do results and not just talk about TSNE. It's really good to, because if we have about 10 more minutes at most, maybe 5 to 10.
Okay.
So, yeah. Sorry.
Sure.
Great.
Okay. Yeah, and I'll be around all day, so you can find me and pester me about things if you like. Cool. And then I'll kind of speed through, but then another popular method that came out, which was zero inflated factor analysis, which is kind of like PCA, but in factor analysis, you say there's different noise across each feature, which makes a lot more sense for genetics, because each gene might have a different kind of variability. It might have different CG content or different uptake, different size reads. And so they're latent. Let's do this real quick so that you guys can then question all my results. And then they add this. And here, Z is latent, X is this in between. They add a dropout term parameterized by a Bernoulli of then right. So they say that based on what we expect the gene count to be, there's some probability of dropout, which decreases as you have more counts. And then we actually observe something with either observation or dropout. And this tries to model sparsity really nicely. There's a lot of other models that kind of built on this where they play around with this link function, use a neural net, use a different functional form, use a GP or something. And we like this dropout modeling, but also we don't love it. And this also still has the same problems kind of as PCA with the linearity and the kind of sensitivity to counts and sensitivity to oversampled cells. So these are kind of the big methods. I know there's been some new ones that came out recently, which I'm happy to talk about after the talk, but I'll just run through some results we got with our model that we're really happy with. So first of all, we're able to scale this on our 16 CPU to 1 million cells. TSNE kind of runs till 10 to the 4th cells, and then we weren't able to get it to converge, at least on our machine. They have ways of you do dimension PCA first and then run to TSNE, but we're just looking at out-of-the-box, unfiltered, unnormalized cells. The zero-inflated factor analysis implementation, as currently, requires a dense matrix in NumPy format, and so you can't go past the memory constraints, at least on the machine we had. So we use this for actually k-means, not k-nearest neighbors clustering. So this is in 3-dimensional latent space. We looked at the pollen data set, which contains 11 known distinct populations from different human sources. And we can see, at least in our latent space, that ours does like a little bit better job of separating some of these known populations. We can point out here, especially the black and the orange for our method, where they kind of mix in zero-inflated factor analysis and PCA, and in the purple and the like dark red all the way on the left. Separates a little better from our method in the other two. We are assuming that this is not our experimental data set, this is someone else's. We're assuming that this pollen data set, the populations that they gave are correct labels. And we can see that if we look at these adjusted RAND scored and normalized mutual information, which are just ways of comparing two different sets of labels and seeing how close they are to each other, our model across dimensions 3 to 9 does a better job of recreating these clusters than the other methods does. We talk about TSNE. TSNE is really great in 2 dimensions. You go above 2 dimensions, and it's really easy to overfit these Z's to kind of perfectly recreate this. And so because there's no global structure regularizer, nothing to ensure that Z maps to Y in some way, it kind of collapses into like this little ball in the around the origin.
And to be fair, it was never intended to be used as a latent representation in higher, in intermediate dimensions. It was intended for visualization.
Exactly. T-SNE is meant for 2 dimensions. I mean, it works sometimes in 3 dimensions, but don't use T-SNE past 2 dimensions is the real takeaway. Everything else, use whatever model you like, but it's all cool. This is with 10,000, oh, the slides are cutting off. This is with 10,000 CD34 plus cells, which are normally a marker for hemopatic stem cells, and we're able to see some really nice separation of behaviors we would expect from hemopatic stem cells. So across dimension 3, we see things like lymphotoxin beta. Oh, no, dimension 2, we see like LTB, lymphotoxin beta, and we see like leukocyte-specific protein, which are kind of indicators of lymphoid activities, which is one derivative of hemopatic stem cells. If we look in dimension 3, going up, we see things that are similar to that we'd expect from myeloid cells, like tyro-BP, other things we see. So what we're mapping here is the actual counts observed on the latent position of the cells. This is Z1, 2, and 3. And it's like S100, A4, and A6, some different genes that generally indicate myeloid activity. And then across dimension 1, we're kind of separating out just general cellular functions with like COX5A. This is CLTA, and then STOMAL2. We tried this on one of those pseudotime data sets. This is from GP Fates. They infected some mice T cells with plasmodium and then sequenced it at daily intervals across time. And we can see that, I mean, all 3 models, TGP, LVM, PCA, and zero-flight fact analysis are all able to recreate the developmental pathways. But based on this mappings, if we have blue as 0 and purple as 7, if I build this minimum spanning tree on the embeddings to kind of see where the nearest neighbor is coming in our latent space, it's a very rough analysis for pseudotime, but it works quickly. R separates the 0 and 7s. The PCA and zero-flight fact analysis kind of separate weirdly between day 4 and 3. And T-SNE kind of finds this global structure, but it's separating like day 4 from day 7, right? And this would change if I change the complexity parameters. So not that T-SNE is bad, but be cautious. And here, actually, we used our ARD kernel. I was able to pick the 3 lowest length scales, or the 3 highest weights, and see this nice kind of 3-phase activity. And when we overlay different marker genes from the consortium that gave this data, we find a really nice separation between 3 behaviors we'd expect to see. And you see something like T-SNE in 3 dimensions doesn't really give a nice representation. PCA works pretty well, but you see that basically higher count genes are just being pushed out to the edge. So we've built this generative model, and we hope it can be used for downstream analysis in like a rigorous way, because the principal model is fully generative, fully Bayesian, and basically nonlinear and very flexible and very robust. We can learn the number of dimensions. We have this really robust residual. You don't have to filter out genes. You don't have to filter out any cells other than like the ones that you don't think are actual cells in your data set, which we think is possible to like preserve information because these experiments are very expensive. And we can use it for all the downstream analysis, visualization, developmental, clustering, et cetera, et cetera. So I'd like to obviously acknowledge, that's my results. I'd like to acknowledge my PI, Dr. Engelhardt, who unfortunately couldn't be here today, the other single-cell members of our lab, Mark, Ari, and Bianca, who's about to present an amazing talk in a couple of minutes, and some of our collaborators, Donna Pear and Vivergev. So thanks for listening, guys.
Thank you very much, Alex, for the kind introduction. Alex and John, thank you for inviting me here. It's a great pleasure to return to the Broad, where I have attended many, many talks that have set the bar very, very high for this one. So I hope this is going to be okay. I am a graduate student, as Alex said, and I'm very interested in problems that arise from statistical genetics in particular. And I like very much to put a statistical machine learning theoretical spin on it. So what I hope you're going to get out of this talk is an understanding of the new species discovery problem. And don't get too excited. We're not going to do any sort of actual new species discovery problem. We're going to look into cell types. Discovery problem. So we're going to understand a little bit what we mean by that in the context of cell type. And we're also going to learn about multi-arm bandits. A little bit bigger?
Just a little bit, yeah.
OK, cool. Is this OK?
That's good. And also, don't go any lower on that right there on the podium.
Sounds good. I was just planning to make an outline here. That's OK. So we're going to learn, hopefully there's a good subset of you that don't know about new species or don't know about multi-arm bandits. For those that are in the intersection and know everything about this, take your papers out because I have some lemmas and results. So if you get bored, you can try to solve them and prove them yourself. This is going to be extremely hands-on. There's going to be two parts to the talk. The first one, I'm going to introduce notation and some simple results. And for any math nerds here, I hope that's going to be fun. It's going to involve some basic statistics and basic probability. So if you have any questions at any time, please feel free to interrupt. And the second part of the talk is going to show some results in both simulation and real data. Cool. So let's start. First, some notation. And Alex, please yell at me if it's still very small. He's distracted now. Okay. So here's some notation. Thank you very much, Archit, for the wonderful single-cell clustering primer. Whenever I say species discovery or cell type, you're going to think about Archit's amazing method and associate a label immediately to it. So for now on with X is going to be label set. I'm going to refer to cell profiles as X. So if I'm going to say X to the n, I will refer to a sequence of n labels. These are observed. And I'm going to use X n plus n to n to denote the set of cells upon observing the first n cells. So this is going to be n plus 1. So the problem that we're going to try to solve is going to try to estimate a particular number. And this number is, if I am to observe n samples from my data and associate some labels to it, how many more new samples should I expect in the future? And this is a very well-studied problem that we're going to get some intuition on. And we're going to understand how to use this problem together with multi-arm bandits in order to address questions related to building a cell atlas in a principled way. So U is going to be a statistic that's going to be dependent on observed and future samples. And it's basically going to be the cardinality of this set. Plus 1, xn. Cool. So I meant this. Cool. So how about, there's any question? I thought I heard something. Okay, I haven't. So let's start with a little bit of history. Does anyone?
So use the number of new ones, just to be clear?
Yeah. So we have access to n samples.
Yeah.
Each of these samples, xi, belongs to the label set X. There are a couple of properties that this set X has. So for example, it can be finite, or if we want to be overambitious, and this is work in progress, we can make it countable. So it can be infinite, but countable. Does everyone know what a countable set is? A countable set is a set in which you can have an order. So it's a direct map between the set 1, 2, 3 of integers to the particular set. So it just means there exists a bijection between the set. And so on to X. So basically, you can order them in a particular way.
What do you mean by label set? Is it like a nomenclature for the cells? Like
yeah, so let's say you do an experiment and you collect n cells, and this dimension d based on how many genes you have. A row is going to be your gene expression profile, and the label, which is going to be x1, for example, xi, is going to be the cell type. So you run your Favela clustering algorithm, you get a label, and that's going to be your cell identity. And whether that is perfect or not, it's up to you. As you collect more data, you get a better estimate of how many cells there are. And the reason for which we don't want this to be finite or countable, because we want to eventually extend this problem to more of a non-discrete density. So having the first step in extending finite to countable is necessary.
But here there's no structure on the label set, right? It's just, there's sort of, you could think of it as like cell type number 1, cell type number 2.
Yes.
So
Yeah.
if eventually we want a continuum, you would also need some notion of nearness or...
Exactly. And today we're going to focus on finite sets. I'm going to briefly mention a Bayesian parametric application using a Pitman-Yor process, but we're not going to dwell on that too much. We can talk about it during the discussion. In this particular case, the other thing that we should keep in mind is, as I'm going to walk you through this computational timeline of how people have approached this problem, we're going to get an intuition exactly on what Alex asked, which is, what is the structure of X that allows us to make any sort of estimate for this quantity? Because there are models for which this estimation is simpler than others. And I will make sure to point those out. So spoiler alert, if this is a Poisson distribution, if the labels are sampled from some sort of Poisson distribution, then you will have really nice theoretical properties that are going to allow you to prove nice statements about you. If you have multinomial model, you can also prove that, but it's a little harder. And then you can move on to putting extra constraints over the label set. For example, in the hierarchical Pitman-Yor setting, you can imagine that you have a big urn. It's basically everything boils down to some sort of urn problem. You have an urn with an unknown numbers of balls of different colors, and you keep sampling from this big setup, and you want to compute some sort of quantity about what the true distribution is. If you assume sampling with replacement, without replacement, so there's a lot of thought process you can have as you're thinking about the problem. Does that answer your question? Oh, cool. So going to the timeline, where does this problem comes from? I'm a really big fan of history, and I like to see how people across years have thought about the same problem and how they added extra structure to make it easier or harder, and why they made it harder, and does that help? So this starts with the. I think I'm having a lot of fun doing that. So, anyways, so this whole story, you're gonna be surprised. There are 4 time points, and they're very spread apart. So 1943 is the first time when such an instance of a problem was posed. Courbet was a naturalist, and he was traveling to Malaya and collecting butterflies. So his trip was very expensive. He had to go at the time, to make a dangerous trip, and had to invest a lot of resources and convince a lot of people to give him money to do such a trip. And he would collect butterflies. So he would make this particular table where frequency refers to the count and not a float number. Species. So he would collect a lot of butterfly and said, okay, there's 118 butterflies that look different, very, very different. So he inferred that from different families. And he only caught one of those. And then there would be 74 butterflies he trapped that would appear 2 times in his population. And he would keep computing this table. And what happened is that he went to R.A. Fisher, who was the statistician in charge at the time. Yeah, so he went to Fisher, and he asked him, so should I really invest in another trip? So you can think about us modern biologists that we have a lot of money to do a lot of stuff, and we would want to think, if I do this experiment one more time, maybe in a different way, do I get different results? Do I get the same data? Should I do it? I already published a paper. I already have data and all these beautiful plots. Disney, probably no. So Fisher thought about it. So Courbet asked Fisher, okay, how many new species of butterflies should I expect that I'm going to see if I go again in my lab? Is this a good representation of my distribution? And Fisher probably, sat at the fireside with some coffee, and then said, like, yes, you should expect this number to be the true answer. And so on goes for the rest of this table. You can imagine that there are more entries. And before I move further, does anyone, once you see this number, do you have any intuition where it might come from? Because I was personally very puzzled when I saw it. Would anyone volunteer?
This is an estimate of the total number of species.
Of new species that you're expected to see.
It's the inclusion-exclusion.
If you were to collect, how many though? the limit of collecting all the butterflies?
That's a really great question. So actually, you're making my job easier because I'm introducing some extra notation. So let's say he collected n samples until now. What he wanted to say, if I'm going to collect the same amount of samples. So he said that if I'm going to collect n butterflies, so let's say in his case, maybe the number of butterflies was around 300 something. He said, if I'm going to go again and collect another 300 butterflies. But hold on to that thought because that's going to be very, very important. So inclusion-exclusion principle, any other thoughts? Any other thoughts? Okay, I will answer this question that right now is almost rhetorical. But so what happened is that this for me is very surprising. It's an alternate sum. It can probably also go negative under some circumstances, and indeed it does. As I'm going to discuss in simulations. So to me, this is a very surprising number, and it only came until it took until 1976, until 1956, where Good and Toulmin, and maybe this is more familiar to you, since there are a number of studies that have used this for identifying variation in SNPs. So it's only until that time. By the way, if I write here, can you see it? Is it
much lower than that?
All right. Yeah, sounds good.
Sorry, I think this was already asked, but what question is this number supposed to answer?
How many new species of butterflies will you see if you are to go again to the same place and collect n more?
N.
The same
There's
number.
no n in the. Oh.
So this is so. For example, let's say here n is 500. So you would keep completing this table until you would reach 500.
Yes, and then the second number is if you collected the same number
Exactly.
again?
Yes.
Okay.
So you go to.
So if you doubled your sample.
Exactly, but it's not in the doubled sample. Is how much you would see in this next in the next setting. So you first observe.
But I don't see that there's no other parameter. I don't see m. You know, so.
Oh, so in this particular case, m would be equal to n.
Okay.
Right, because you're seeing samples from n to 2 times n.
So R.A. Fisher's result is about doubling your sample.
Yes.
Okay.
Okay. Yeah. Sorry, I think I misunderstood your question. I thought you wanted to say that what would be the expected number if I would double my sample. So for me, I thought you mean that m is equal to 2 times n.
Oh, sorry.
Yeah.
So you're saying if you went back and collected the same number of butterflies you already collected, but new ones,
yes.
How many more fresh species would you
exactly.
observe in expectation?
Exactly.
And that's what the,
yes.
That's that number.
Yeah, exactly. Is that clear to everyone? Sure.
So how many species have you observed at this point?
In this particular case, you observe 500, and there's more entries here, because this doesn't sum up to 500, hopefully.
Wait, what?
Okay, let's take an example. How about we take an example? Let's say you have n is equal to 7, and you're collecting letters in the alphabet. And let's say your x7 is bananas. No. So here, the way you would say x1, how many letters appear only one time? So here, like there's only there's 2 letters. There's B and S that appear 2 times.
This x1 and that x1, those are different, right? They're the same. I just don't want to use the same.
Okay, no, I'm consistent in my notation. So XF is a sequence of letters. So here the alphabet is just the letter. So B, A, and so on.
Right. So x1 is not 2. X1 is B.
Yeah.
Oh, I'm so sorry. Okay, yeah, you're good. Thanks for pointing that out. So I was about to introduce a new term here, which is called prevalence. So phi is called prevalence, and it basically refers to the entrance in this table. So this is phi1, this is phi2, phi3.
So it's the number of labels. Phi is the number of labels that are observed exactly I times in your data.
Yep. Yeah. So here there would be 2, and then phi2 would be how many things appear 2 times. So there's the letter n. So this is 1, and to answer the question about the species, imagine you kept looking at other letters. You know, this is what you've noticed so far. This is the type of butterflies. And then this would be XS plus 4 because you get 4 more letters, right? So this would be an estimate, let's say, I don't know, this is ZT. So now it would be 7 plus 7. And, yeah, so basically here the new species observed in this letter are X, Y, Z, and T. So there's 4. So in this particular case, U of X7 and X14 is equal to 4. Yes?
To analogize to population genetics, this is similar to given a histogram of allele counts and a population size, exactly. How many new alleles do we expect to find if we sample again?
Exactly. Yeah, that's exactly the application I was referring to when I was saying that this particular Good-Toulmin, and I think that's some work by Ionita Laza.
So you're talking about discovering polymorphisms?
Yeah.
Yeah. So actually, this is one of the applications that I think a couple of years ago, this particular estimator has been used in that context. Yeah, we're using it in a slightly different context right now because we're using it for clustering, and we're using it in the setting of multi-arm bandit, so it's an online problem. But that's exactly the reference you should have in mind when thinking about it. Thank you. By the way, I forgot to say this. So this is all work based on a preprint that's available on the bioRxiv. I'm trying there to give a fair treatment and history of all the papers that might have used any variation of these methods. If you find something that you think it's not properly cited there or refer, please message me. I'll be very happy to include it, compare against it, and everything else. So actually, thank you so much for bringing that up.
Have you cited Turing, Alan Turing?
Sorry?
Have you cited Alan Turing?
For
German tank travel?
What's that?
Because it's a German tank travel?
No. So one of the first papers actually written on this problem was by Good and Turing.
So that's a different, actually that's a good, yeah, so the question was, did I cite Alan Turing? Because there's another estimate which is called Good-Turing estimator. It's not the same estimator. It has a different interpretation, but we can talk about it during the discussion or afterwards. But we do cite that paper. It's a very well-known estimator, but it solves a slightly different problem with less structure. So we're not going to talk about it. But actually, this is very important. Do not confuse this one with the other one. They're very related, but they do different things and have different statistical properties.
Most people haven't heard of either, so
yeah, so it's good. Yeah. Well, thank you very much for your question, though. Okay, so are we good on the new species labeling? Is everyone clear on the fact that in this particular case, the new set of labels is 4? Are we? Yeah. Oh. So I will. Let me see. I will erase this notation, but if at any point I'm inconsistent, please spot it. And it's going to be great. Sorry for the noise. I think just cool. All right. So here's what Good and Turing proposed. So they proposed a way to formalize the Fisher estimate in the following way. So John and Alex asked about whether the sample is doubled. So what Good and Turing said is, what if we're actually considering M in general, and we're characterizing M as a constant T times N? So the point is that what if I feel that my sample is very diverse, and I want to invest T more funds in sampling? So in the Fisher case, T was equal to 1. And Good-Toulmin are thinking about this problem in a little more generality. So the estimator that they came up with was the following one. Where this term is the prevalence that we talked before. So that was very convenient for me because I wanted to have a toy example. So you can see it's an alternating sum. And you can see that if you just look at it, you can see that it can have both really good properties, but also really bad statistical properties. So what are those? Well, what they managed to prove was the expectation between the Good-Toulmin estimator and the estimator of new species in this particular context is bounded by this number.
So the second U is the count, right? Second U is the actual random variable, which is the number of fresh ones.
Yes.
Okay.
Yeah, so the first, no, the U is not the count. The U is the best estimator that you can possibly come up in theory. So yes, it is the count. It's as if you're an oracle and you saw the sample before. And you tell me you're going to see this amount of new things in the sample. Great. So, so the Good-Toulmin estimator is just this particular function that for t is equal to 1 reduces to the RA-Fisher estimate. So why is this result really nice? Well, in the case where t is smaller than 1, then you can think about settings where you're saying, OK, my Good-Toulmin estimator is more or less around u, but if t is much bigger than, it's bigger than 1, what's going to happen is that this particular sum is going to be dominated by the t to the I term. So what they also managed to prove and think about was that the variance of UGT is tends to infinity as you collect more samples. So that if this quantity was a function of as n goes to infinity, then this variance would be very large. So that was not really good for them because it was very hard to estimate, and it had this undesirable property.
So in the case Fisher considered, it tells you nothing, right?
Yeah.
Okay.
It's what do you mean it tells you nothing?
Well, if you collect n more, if t is 1, then you're bounding this thing by n, which is like the largest possible number of fresh species you could ever see.
Yeah, well, so here's the thing with this particular estimate. This is a result in distribution. So actually, you can get better bounds in general. This is just yeah. Actually, in other papers, they managed to show that this bound is much tighter. Yeah. So, but that's a great observation. So basically saying that if t is equal to 1, we actually don't have any sort of concentration in expectation. And in a classical machine learning sense, or where you would want this to be e to the negative of something that goes to 0. So it's not a concentration result in that sense. It's just illustrative in the sense that, OK, this term is very unstable, and you can't say much about it. Actually, this problem did not receive much attention until 1976. You can see there is a big gap. And in 1976, Efron.
Bianca, maybe you should move over because I don't think people can see because of the podium.
I see. Okay. Sounds good. Yeah, I will get rid of this. Meanwhile, if there's any questions, I can attempt to answer while I do this very menial task. All right, so in 1976, Efron found a new application for this problem, and he was looking at the works of Shakespeare, and he was saying, if I read a couple more works of Shakespeare, how many new words should I expect to see? So this is the application that they looked at. They didn't really have large-scale genomics data back then, so it was hard to think about other problems and applications. So I have it very easy. So what they proposed for dealing with the instability of this term was a very simple variance reduction technique. And they simply said, we're going to truncate it. So then they looked at estimators that had this particular form. So they proposed a new class of estimators. Which is a linear estimator in the prevalence, which we defined before. And the particular property of this estimator is that the linear coefficient term corresponds to this probabilistic quantity. So if you are to decompose this term, what you're basically going to have is going to be a sum from for some k in the integer set. So what you're going to have here is going to be a sum over k terms, which if you just expand the binomial, you would get this not particularly friendly term, but don't worry, we're not going to use it that much. It's just for completion. And for the case where the index is going to be bigger than k, these terms are going to be 0. So what does this do intuitively? It's basically saying, I'm going to fix a k, which is a natural number. After I see k terms, I'm going to make all the prevalence terms 0. So that's what I mean by truncation. And not only that, but if my k is very large, I'm going to make sure that I balance this ugly term that I don't like with some sort of weighting, such that I make it less and less probable as it gets bigger. And this is kind of a classical technique that people use now, even in optimization when there's an alternate series and they try to bound or approximate. So what happened with this is that it's easier to compute. So it's easier to compute. In application, it worked better than the original Turing estimator. But the not so nice thing is that there was no theoretical guarantee. And while this might not be so sad for a lot of us, we have data, we don't always have theoretical guarantees on our algorithms. But what the particular problem with this was kind of the problem with k-means, in a sense that we didn't know how to choose k. So there's no theoretical guarantee. We don't have a principled way of choosing what k is. And you can do what you do in k-means scenario. You can do some sort of you can try different values of k, but you don't have an idea of an elbow that would tell you, okay, for this particular k, I will stop and choose 5 clusters rather than 10 clusters. It's absolutely no intuition in this particular formula. And no such intuition came until 2017. I'm going to write it here, 2017. And there's a beautiful paper by Orlitsky and co-authors where they actually managed to prove that a class of estimators related to this have actually really nice theoretical properties. So I would, for time reasons, we can talk about this during the discussion, and I can point you also to the review that they wrote, which is really beautiful, and actually one of my favorite papers, I think.
So they made it more complicated, but it helps.
No, it wasn't more complicated. They actually looked at general estimators here, and they chose for particular distributions. They showed that this estimator relates to that distribution, and they have theoretical results on the convergence of the estimator and unbiasedness. So they didn't necessarily make it more complicated. And a lot of history of this estimator is not about making them a little more general and showing that in that general class it's easier to prove things. Yeah. So, but yeah. All right. So one of the things that we don't have at this moment, we don't have intuition of how did they even come up with this. So in the next 5 or 10 minutes, I'm going to spend some time picking a very specific model, and we're going to get our hands dirty with this particular estimation. So one of the simplest statistical models, so intuition. So one of the simplest models is the Poisson model. And it's been a favorite of statistical genetics community and population genetics community. So we're going to take some liberty here in how to define it. So we're going to say that n and m are some integers, and we're going to choose the number of things we're going to observe is going to be Poisson with parameter n. And sorry, I meant to say that this guy is an integer, and m is like Poisson with some parameter m. And basically what we're going to do, we're going to observe xn plus m samples from an unknown distribution D, where from an unknown distribution P, which is the probability distribution over the alphabet space. And we're going to say nx and n prime x are the multiplicities of a label small x in the first n samples, and this one is going to be in the next m samples. So here's something nice. The expectation of, if we want to think about how many new species we're going to expect, we can write this really nicely as a sum over all the labels in our label set. And this is going to be an expectation of one, like an indicator variable. So basically, like, if I want to rewrite this theoretically, it's basically a sum over all the things in the alphabet and all the things that do not occur in the original set times the expectation of things that do occur in the next set. So this is if I want to formalize.
What is nx?
Nx. Nx is the multiplicity of label x in the first n samples, and n prime x is the multiplicity of x in the next m samples. So if you remember the bananas, you would say that if we look at A, n of A would be, in the first 3 samples, it would be 1. And n of A in the next 4 samples would be 2. It's as simple as that. Cool. All right. So if we also look at, if we define lambda x to be the parameter.
It's too low again.
It's too low?
Yeah.
No. I had.
To the right a little bit.
Okay. Here? All right. Thanks. You guys have a lot of experience with like all the possible angles. It's like good segmentation skills. All right, so basically what's happening here is that we're having, if we say lambda x to be n times px, where px is the probability of seeing label p, and then is this parameter here, you can trust me to know that nx is actually distributed as a Poisson with parameter npx, which is equal to a Poisson with parameter lambda x. So why these particular things are very helpful, it's because now we can write all these estimators in a very convenient form. And I'll show you what the convenient form is in a moment. In particular, we can write, as I said, that the expectation of u is equal to these 2 events. So it's a sum over the things we have, all the labels in our set for which we haven't seen them in the first n samples times the indicator variable that we have seen them in the next sample. And due to properties of expectation, we can move this outside. And since these 2 are independent, due to the fact that we assume this is a Poisson model, this looks like this. And by the way, since I'm writing and I'm not looking, please feel free to interrupt at any moment. All right. So we actually know what these terms are because we made the assumption that these things are Poisson. So then this is a sum of lambda x times like this. Why is this? I mean, technically, we would have a more complicated term. So we would have lambda x. Then there would be the lambda x to I. And then there would be an I factorial, normally, for the probability. But when I is equal to 0, then that's a really nice term. And this is all that remains. Yeah? Because just 0 factorial is 1. And when I is 0, this particular term goes away. So this is what this number is here. And this other number can be written as 1 minus. Oh, and the other thing that I took the liberty here is to say that if I have the expectation of an indicator variable, then this is actually just the probability of that particular set. Yeah, this is the other thing that I did here, and that's why I don't have any integrals or any sort of sums. Cool. So since this is the opposite event, this is going to be 1 minus e to the minus t times lambda x. And where does this t comes from? This t comes from the fact that, again, this is m over n. I'm just pulling it into this lambda x constant. All right. So now this looks much friendlier, and we can do just a little bit more mathematical manipulation. And what's going to happen, it's promised it's going to be really nice. We're going to do a Taylor expansion around this function. So what's going to happen is that we're going to have here a set of terms, let's say over j, and this is going to be minus t to the j times lambda x j over j factorial. This is lambda and not x.
This is j greater than 1.
Yeah.
Greater than 1.
To infinity. It's just
greater than or equal 1.
Yeah, and greater than 1. All right. So we can also move these terms around to make it a little nicer. So what we can do is, we can just pull this term, and I'll move around for people on this side to see as well. We can just pull these terms around a little bit, and what we can do is this is sum over x e to x. So what is the nice thing about this right now is that, does it remind you of anything you've seen so far? Does it? No? Oh, it means I'm doing a horrible job.
Good job. It's the puzzle.
Yeah. It's a puzzle. Well, I'm going to tell you that this actually is, so there's, I forgot the minus here.
This is the estimator you had before.
Yes, it's the estimator we had before. In my evil plans, all this whiteboard would have been neatly organized and you could refer back to it. But maybe in 5 years, when I give a job talk, that's going to be all figured out. And if I... Yeah. Yeah. Anyways, so actually this is exactly true. It's the estimator we had before. So this is j is equal to 1 to infinity of minus t over I times the expectation of phi I. Because remember when we defined, I don't know, did I have defined this anywhere else? Yes. Yes, I did. Okay. But just to remind you, remember that the expectation of phi I is actually, so the expectation of the prevalence is that basically you go over your alphabet x, and this is the case that 1 and x is equal to I. So basically this expectation is that the expected number of terms that occur I times. So again, because this is a Poisson random variable as per assumption, the sum, so this is by additivity, of course. So we can say this holds here. And now this is a Poisson variable, so it actually is exactly this term here. Yeah? Yeah, so now you guys can feel like Fisher. Basically, as a lot of the statistical models in that time, people would always start with the Poisson assumption when they would have to work out these details. So this is likely where the estimate came from. So any questions at this point? Are we ready to look for some experimental results? Yeah? You're like, yes, I want to be done with this. No.
So under a Poisson model, Fisher's estimate has the correct expectation.
Exactly.
Is that correct? Is that
yes.
right?
Yeah, which to me was very surprising and quite beautiful.
So that's a simple theorem. That's just true.
That's just true.
Okay.
Yeah. So when I saw the results first time, because nobody really proves it very carefully, A lot of these derivations can be found in this really, really nice paper. Yeah, so so you might ask, okay, what is the challenge now? As it was previously mentioned, some work exists in the context of looking at variation in one particular data set, but what it doesn't exist right now, I mean, it doesn't exist. There have been some recent ways of looking at this problem in a different way. What happens now if we have multiple islands? What happens now if instead of having one island where you go and collect butterflies, you have to choose between 100 islands? So this is kind of the motivation for this project. And what I really want and what I'm very excited about, in the past few years as a graduate student, I developed some interest in process. I'm very interested in end-to-end training of various models, and I'm very interested in end-to-end science. And what that means to me, it means that you have a process in which you model your data collection problem, you run it in some algorithmic fashion, you get some results, and then the results should inform your choice of next data. And I'm looking at this as an iterative process. And in some cases, it might just be very nice to collect a lot of data, millions of cells, and get an estimate, and that might be enough. But I argue there are some cases where that is not possible still. And we're going to talk about this in more details in the next few minutes. So the particular setup I have in mind, imagine you want to build a cell atlas, and you can collect many points from many different regions. Or imagine you have a tumor, and you want to collect a spatial location from this tumor, and you want to have single cell resolution. So in this particular case, the three colors correspond to three different tissues. You want to collect some data, you cluster your data, and then you want to readjust how much budget you spend in a particular region. Maybe there's a region that's not very interesting, so it has very little diversity. So you can think about this. If you're an immunologist, maybe you can think about receptor diversity and all sorts of questions related to that. So you might want to really not focus on one region at all and readjust the budget that you're interested in investing in that particular experiment. And you can think that this is very relevant maybe for 3 regions. That's not really a problem because you can do it in 3 different labs. You can synchronize the groups. But if you have things that are a little more complex than that, let's say you're looking at FACS sorting and you want to see what's the association between a FACS profile and the gene expression. You have around 30 markers, then you have 2 to the 30 combinations that you might want to sample. You might need at least 100 samples in order to see any sort of clustering. You can see that that number can become really large. So what we want a framework in which to build some intuition of how to explore this very large space. And this tool comes from reinforcement learning. This originally is a statistical problem that relies on a very unfortunate metaphor, and that of a gambler going to a slot machine. One-armed bandit is also referred to a slot, a slot machine. So basically, the idea is that you want to, you have access to a couple of such bandits, and the reason for which they're called multi-armed is because there are multiple of them. So what happens here is that as a gambler, you want to spend your time playing at each of these machines, and each machine gives you a particular reward. And you have some mental model of what the reward can be from each of these machines. And if there's some particular noise, you might have problems prioritizing and choosing what's the best one. And there are many, many types of bandits. There are contextual bandits, there are correlating bandits. If you look at any of the machine learning conferences, there are dozens of papers on variations of bandits. One of the particular ones we're going to use to illustrate the benefits of the Good-Toulmin estimator is a contextual multi-armed bandit, where the context refers to the gene expression profile. So in a contextual bandit framework, what you do, you have a couple of arms. So in our case, the arms are the islands, or they can be the regions that you're sampling your data from. And you want to select the best arm in which you should invest a budget, or you want to select a group of arms that might help you reallocate the number of cells you want to collect. And what is the challenge here? The challenge is that you really want to be able, I'm sorry for this slide, I think it is very technical, but bear with me for a moment. The goal here is to really learn the underlying parameters of each of the bandits. such that you can choose at each step the one that's likely to give you the most reward. But
can you translate the Bandit thing into the cells?
Exactly. Yes, I think that's a great point. I think I spent a lot of time on this, the math part, but I think there should be some. So here's making the metaphors a little more precise. So let's think about, there are three main things that are important in the multi-arm bandit. So there's the arms, there's the parameters, and then there's a reward. So at each point, I choose a bandit, I get a reward, and using that reward, I want to update my parameters and choose what would be the best arm to sample. So in this particular case, arms is a region. It can be a tissue. It can be an island in the butterfly setting, but it can be a setup of developmental stages. So it can be time. Let's say you want to see how a particular population of cells changes across time, and because you cannot repeat the experiment in that population, you want to grow the cells for a particular time, and then you want to see at which time to stop to get a better diversity. Okay, cool, I have 5 minutes. The parameters in this particular case are the parameters of the algorithm you use. For example, any sort of clustering parameters that you want to update in an online way. And the reward, this is actually a very tough one. The reward, and this is one of the main challenges, is that you really want to be able to use this developmental time, tissue regions, in order to maximize the budget across the samples. So the reward is actually going to be the number of new cell types, for example, new cell types. So you want to have a measure, cell types. You want to have a measure on how surprised you're going to be if you repeat the experiments and see something very different. And that's going to be your reward. If you are to query one arm and with those current parameters is going to be telling you that you're going to be very surprised, then you should pick that arm. And actually what the bandits do, they balance between two things. And those are exploration and exploitation. The exploration means you want to sample your space to get an overall baseline intuition about your system. And the exploit part relates to the fact that once you know which one is the most diverse and the one is least diverse, you know how to allocate the appropriate resources in each of them. And the way we approached it in the context of the mouse cell atlas, which is a data set collected and published at the beginning of 2018, it's a collection of over 1,800 cell types and 40,000 cells. Actually, this is the amount of cells that were curated and we ended up using. So the idea is to frame this sequential experimental design as a multi-arm problem where the number of new cell types is modeled as a Good-Toulmin estimator. So you kind of, actually asked me, you anticipated the type of metaphor I wanted to explain. So again, arms are the scenarios of interest, contexts are the cells, and the reward is the cell type diversity. So we already went over how to model the diversity setting. You can see here an example of an algorithm that you might want to do. K is the number of regions or arms, and U is the estimator you got very used seeing. But in this particular case, you see a U for U of J indexes how many regions you have. So basically, you have an estimate for U for each particular region you consider. And at each moment in time, you're trying to allocate cells according to this probabilistic scheme in which the reason for which there is this discount parameter alpha here, it's because sometimes the estimator might go under 0, and you also don't want to completely lose interest in a region that might seem not diverse at the beginning. In a simulation
that's
results...
exploration. More
yes.
alpha is more exploration.
Exactly. Actually, that's a great point. So if you have a very, very big alpha, that's exploration. It's kind of going to make your distribution very uniform. And if alpha is very small, you're saying, I'm really trusting this estimator. I'm just going to find the thing that really exploits most. Yeah. So in the simulation results, this is a simulated population with where there's a subset of around 100 arms. Half of them are have a very low diversity, and the other ones have a lot of populations. An Oracle strategy is something that's allowed to see in advance. So I'm collecting the data. I see how many cells I do, and then I use the appropriate estimator with that number. GTTS is the proposed method. GT stands for Good-Toulmin and TS from Thompson sampling. The Thompson sampling has to do with the choice of the probability distribution and how you choose the next arm. If you want to hear more about Thompson sampling and UCB algorithms, you can stay for the discussion, and I can give you an extended tutorial on that. And the delayed abundance case here is the setting in which you don't choose one particular region at each moment in time, but you can select a subset of them. So that's why you're doing a little bit worse. To finish with some results, we use data from the mouse atlas, and we aggregated them in terms of developmental stages. The number of cells is very different, and the diversity is very low in the embryonic stage. And what you can see is that we actually recover, if you have a random strategy where you don't care what you sample from, you might say you get pretty good results, but if you have a limited time, you can notice that after a particular trials, you can get up to 10 new clusters. So on the y-axis, you have cell types or how many things you uncover, and you can see that even if you had more and more cell types, you're not going to really converge as fast with the random strategy. So if you really care about uncovering rare cell types in less time, you still might want the advantage for this particular method. Even more compelling reason for which this actually works is that if you knew from before what are the best samples to choose from, and this is the oracle on the left, then you would invest more time into sampling cells from the adult. Again, this is a conceptual case. The problem can become more interested with different applications. You notice that in the random cell exploration, you spend equal time in each of the developmental stages, whereas in the other algorithm, you have some time that really approximates very well the distribution that the oracle would give you. A work in progress. I'm working with Federico, Ferrari, and Stefano Favaro. We're working on, we already have the results. We're hopefully, stay tuned. In a few weeks, we're going to have a preprint on the hierarchical Pitman-Yor for species discovery. And in submission, if you want to read more about this result, this is on bioRxiv, work with the fantastic undergrad Feng, who was my mentee in the past year, and she's going to be on applying to grad school in a year. So she's amazingly talented and did a great job. For a broader discussion, the goal for me is to move from this discrete setting to a continuous setting. What we really want to get, we want to not be able to have a very discrete choice of clusters, but what we want to make these estimates in a manifold like setting, because that's something that's going to be much more interesting from the point of view of looking at trajectories and diversity. The other goal is to extend this work to other experimental settings, such as fact sorting or any sort of application. I want this to be an ongoing dialogue. If you have ideas for applications, I'm proposing to you that having some sort of statistical methodology to inform the experiments you do can be beneficial. And I would be very excited to hear about the data sets you have and what you want to work on. I would like to spend some time acknowledging Stefano Favaro from University of Turin and my collaborator Federico Ferrari from Duke University. Also Marco Battiston for being kind and sharing his code and expertise and talking about his paper, which is very similar, and I invite you to read it. Last but not least, I want to thank the Beehive group, which is Barbara Engelhardt's group. Everyone has been very supportive. I'm at the end of my PhD journey, and I had a terrific time. If anyone is an undergrad here and is looking for new place, you know, I think you should really talk to Barbara. She's amazing. And everyone in the group is very supportive, and I'm very grateful for all of them. Thank you so much. If you have any questions. Thank you.